Microscopes, sensors, spectrometers, field studies. There is a sea of data waiting to be worked on in physics, chemistry, and biology labs.
We are the group of students at NYU who brings benefits of artificial intelligence to science labs. As of now, in the third semester of our operations, our group has 8 teams developing solutions for collaborators from various fields of science. Each team forms out of 3 to 5 students with complementary expertise in AI, research methodology, and industry-type project development practices.
While AI is heavily used in commercial companies, there is still only limited use of it in scientific labs.
We develop operational structure in which students learn by gaining experience with unique datasets and problems, while scientific labs get a boost in leveraging AI. Organizational principles come from authentic research education methodology under the guidance of the group mentor Dr. Sergey Samsonu. In the matter of fact, we believe this can be replicated across universities to address the AI shortage in scientific labs across the country.
We learned how to find and set up projects, how to develop a solution actually useful in a scientific lab, and how to package and deliver software.
Student leadership experts make sure every member of the group has sufficient resources and help.
We learn from each other. Twice a year, we present our work on group-wide presentations. We collaborate, we share. Here are some of the projects our group worked on recently.
We help model the spectroscopic data of fluorescent nanodiamond particles using advanced statistical methods. Our collaborator, Dr. August Sendrova from Advanced Nanotechnologies, stated, "It was exciting to see how within a short period of time, the team grasped the essence of the challenge to handle heterogeneous spectroscopic data of fluorescent nanodiamond particles and started development of a tool to process and represent meaningful scientific results. I could clearly see that this tool will be beneficial for academic and industrial users of material with heterogeneous fluorescent properties."
We automate description process of scientific figures in papers. In fact, we are developing novel dataset for computer vision. Here is what Dr. Jonathan Bragg from Allen Institute for AI stated. The VIP team made tangible progress towards building tools for making figures in research papers more accessible via automated alt text generation. This problem has high potential for impact, as most figures lack alt text and prior automated approaches do not target alt text. The team operated very professionally and tackled a range of problems, including close read of prior work, baseline model training, template-based dataset generation, and even created a command line interface to help other researchers generate their own datasets. We identify patterns in particles' motion using unsupervised and supervised learning techniques.
Dr. Romana Shirhagel from the University of Groningen stated, "The VIP students helped us develop an AI solution to identify specific areas of sperm cells. We are hoping that this will help us in the future to research how we can help infertile men to finally fulfill their dream of having a family."
We work on classifying vaccine reactions utilizing classical machine learning.
Dr. Christine Vogel from NYU stated, "From the beginning, I have been deeply impressed by the professionalism of the team. Our interactions really were as if this is a startup offering a service to me. While I'm hoping we'll make even more progress within the project, the first results have already been very exciting. Within just a few weeks, the team parsed a complex human vaccination dataset and was able to improve upon some predictions that we had tried to make earlier. We identify sleep patterns of the mice by looking at the time series data. Here is what Dr. Chowdhury from NYU Abu Dhabi has stated. We are currently collaborating with the VIP team in order to get help from their expertise in performing more detailed analysis on our EEG sleep data. Their analysis has the potential to lead to the development of a predictive tool of resilience or vulnerability to stress, which has broad implications in our society.
Trying to improve malaria diagnosis by identifying the most indicative symptoms of the disease.
We train neural networks to automate image classification. Professor Christine Ganselas from Center for Genomics and System Biology at NYU stated, Our laboratory studies embryonic development in a simple animal model system, C. elegans. One roadblock inherent to large-scale experiments of this type is the ability to analyze the image data without relying on manual classification by human observation, which is extremely time-consuming and prone to errors. The VIP group developed a machine learning model to classify each individual image into one of these categories. The initial results are very encouraging and indicate that their model has the potential to improve our scoring by providing estimates for embryonic lethality in a vastly reduced time frame.
And we are building a network of collaborators to have future projects for upcoming semesters.
AI for scientific research, building better future for science together.